molecular biology. First, we'll look at the chromatin structure. So, what is chromatin? Chromatin is the condensed form of DNA. So, whenever we know that DNA is a very long structure, it is a very, very long structure, and the nucleus or the nucleolus is a very small structure. So, this DNA has to fit into this little nucleolus. So, that would require its condensation. So, what it does, it forms nucleosomes. How will it form nucleosomes? There are certain histone proteins, circular proteins, and DNA will loop around and it will keep on looping and will form nucleosomes. And these will appear as beads on a string. H1 protein is a protein which stabilizes the structure and it is connected to each octamer of histone. Now, during mitosis, chromosome formation takes place, and this requires the condensation of DNA and it's transferred from the parent nucleus to its daughter nucleus. So S phase and S phase. So during the whole process of mitosis, we have a phase called S phase. And in this phase, DNA synthesis as well as histone protein and synthesis occurs. So first let's look at its structure and then we will talk about its peculiarities. So this is our DNA double helix. We know how it looks like. And this is the histone octamer. These are the four histone proteins present and twice they are present, so it is an octamer. And the DNA will loop around this histone octamer and it will form a nucleosome. And this is a string. We can say that these are strings. These are the beads on a string structure, which we can see here. And this is the H1 histone protein, which is stabilizing the histone structure, the nucleosome structure. Now, let us look at the types of chromatin. This entire structure. You can see this. This entire structure is the chromatin, and you can see that it will form a chromosome, and it will help in the cell division during mitosis. So let's talk about what two types of chromatin: heterochromatin and euchromatin. So heterochromatin, HC, HC because it is highly condensed. That is why it is called heterochromatin. And euchromatin is true chromatin. It is not very uh, highly condensed. So it is not highly condensed. Now, euchromatin is also known as true chromatin. Now, if we look at its structure, then heterochromatin will appear darker in color, and euchromatin will appear lighter in color. Let's see the diagram, and then we can see this entire structure is our nucleolus, this entire structure, and this dark area is heterochromatin, and this lighter area is euchromatin. So this is the highly condensed area, as you can see, but that is why it is dark, and this is loosely arranged, as you can see. This euchromatin is loosely arranged in the entire nucleolus. So it is true chromatin, highly condensed chromatin. Now there are two processes that take place. One is known as methylation and the other is known as acetylation. So methylation is also of two types. It can occur in DNA and we call it DNA methylation, and it can occur in histone and we can call it histone methylation. So what occurs in DNA methylation? The expression of DNA is changed. How we express DNA, that is changed, and it does not change the sequence of DNA without changing sequences. And in histone methylation, reversible suppression of transcription takes place. What will happen? Transcription will get suppressed. And in acetylation occurs in histone. So in histone acetylation, transcription will increase actually. Methylation and decreases in histone methylation, but in acetylation it increases, and in deacetylation it will decrease. Transcription decreases. So this was about the chromatin structure and heterochromatin, euchromatin, everything that we discussed. Now we'll discuss about nucleotides. So what are nucleotides? First of all, there are a few nitrogenous bases that are present. What are these nitrogenous bases? Purines and pyrimidines. Purines have two carbons in them and pyrimidines have one carbon in them. Purines are adenine and guanine, and pyrimidines are cytosine, uracil, and thymine. These are pyrimidines. These are these sorry, these are not two carbon structures. These have two rings in them. Purines have two rings in them, and pyrimidine has one ring in its structure. So these are all the nitrogenous bases that are present, and we'll see their structure for a second. So this is a purine, adenine guanine. This is their structure. So we can see there are two rings. So first of all, when we have to memorize its structure, we can memorize that we have to draw a hexagon first, and then we can draw pentagon. And in the hexagon, we'll start numbering from here. This is 1, 2, 3, 4, 5, 6, and then 7, 8, 9. This is its structure. Now we will see its origin, like which carbon comes from which structure, which nitrogen comes from which structure. So there are nitrogen is present, so we are memorizing it. So just for the memorization purposes, you can see two nitrogen on tops, two nitrogens on top, and two nitrogens at the bottom. So the nitrogen which is present in the first position is given by aspartate, and these 3 and 9 are given by glutamine. 3 and 9 are given by glutamine. 4, 5, 7 of the structure. You can make a triangle like this and you can memorize it. This 4, 5, 7 is given by glycine. Top is given by CO2. And these 2 and 8. 2 and 8 are given by tetrahydrofolate. These are the carbons which are given by tetrahydrofolate. So this is a complex structure to memorize, but if you correlate the numbers and learn, then you can easily learn that you make a triangle and then glycine will contribute to these structures 3 and 9. No, this is a multiple. So 3 and 9 given by glutamate. 2 and 8 again, we can correlate these numbers. 2 cube is 8, 3 square is 9. So we can learn it that way. And this is the structure of a pyrimidine. It is a very simple structure, as you can see. Cytosine, uracil, and thymine have these structures. It will just constitute one hexagon, and here we'll have nitrogen. Here and here we'll have nitrogen, and this area will be contributed to by aspartate, and this will be contributed by carbamoyl phosphate. That is it for the structure of pyrimidines. Now nucleosides. What are nucleosides? Nucleosides are nitrogenous bases plus ribose sugar. So when we take one of these and we attach it to a ribose pentose sugar, then we will get a nucleoside. And what are nucleotides? Nucleotides are nucleosides plus phosphate base, phosphate moiety attached to it by a at the fifth position phosphate will get attached. And when will it become a nucleotide? It will become a nucleotide. When this will happen, it will become a nucleic acid. When two phosphate groups or two nucleotides are joined by 3,5 phosphodiester linkage, that will become a nucleic acid. So nucleic acid will be RNA and DNA. That is what formation we had to see. And DNA has phosphate linkages, right? It has a phosphate backbone. The DNA double helix structure we'll see again. See, and then we have again reached a full circle and we formed DNA. So these are phosphates. Right, so it is what DNA is negatively charged and histone proteins are positively charged because they have lysine and arginine in them. So this was it for the basics of molecular biology.